It's a real pleasure to be here again after a number of years. It's so inspiring to be able to talk a bit about my work in the midst of such an incredibly creative group of people. I'm going to go back filled with inspiration and picking up the brushes again. So I'd like to talk a bit today about the work I've been doing for the past 20 years or so. And I'm going to start with this question right here. It's a big echo. I feel like the voice of God coming down here. I'm going to start with this question. What got you interested in science? So, you know, I was a classic little nerdy junior scientist when I was a kid. I had the bug collections and the plant collections and the microscopes and the telescopes and chemistry sets and all that kind of stuff. And looking back at that, the thing that got me started at the very beginning was this series of wonderful little books. Okay? I'm sure everybody of my generation had these same things on their shelf. And you can see this one over here, the insect one, which got so much love over the years that it lost its cover. And I'm so glad to see that you can still buy these little books too. They're still on the shelf. So, I mean, even after all those years, they're still inspiring people about science and natural history. And so looking back at some of these books, I kind of looked back at some of these a couple of years ago when I was setting up an AMI talk, and tried to see what it was that was so effective about these books. And so there's a few things on these pages that are just absolutely perfect for that kind of young reader being introduced to natural history. So these pictures up at the top show me exactly what that insect, what it's going to look like and where I'm going to find it. Right? It's in its natural environment. I can almost see if I go and find that particular tree, I'm going to be able to find this bug. And that's paired up with a really nice, short, descriptive text. Really easy to get in and out of. You know, you're not going through a whole bunch of jargon, just giving you just what you need. And then the most important thing, of course, is this little map down here in the bottom. That tells me that I'm going to be able to find, since I live in California, I'm going to be able to find one of these bugs, but not the other one, unless I talk my family into taking a trip to the East Coast. And actually, these the 17-year cicadas, whatever, have been on my bucket list for years to see, right? Always being a West Coast person. Finally got to see some a couple of years ago, which is a big thrill. Okay, so looking at my own work since then, I find that I've kind of always built on the same approach. And so I'm kind of doing the same thing, but trying to do it for a different domain of knowledge, which is this domain of cells and molecules. Excuse me. I've kind of worked on two big overarching projects over the past 20 years or so. One over here on the left is this idea of trying to give us a view of the molecular structure of living cells. How molecules are packed inside of cells, how they interact to perform all the tasks of life. And then the second big goal I've had is this zooming in on one of the molecules in that environment and trying to tell stories about its structure and how that relates to its function. I'm trying to make that whole mysterious, invisible world a little more familiar. And so today I'll just talk about those two different projects and how I've approached them from the visual side. Okay, so I'll start with molecules. So the challenge of drawing pictures of molecules is a lot of fun, because of course these are smaller than the wavelength of light, so you can do anything you want. Okay? There are a bunch of limitations, of course, because you want to keep it true to the science. The scientists are giving us atomic structures so they can tell us where every atom is of those molecules. And they can tell us what types of atoms there are. You know, some are charged, some are not charged, some like to be in water, some don't. But after you have that information in your hand, you can do anything, okay? And so it gives you a lot of freedom, and you can fall into a lot of traps as well. So I'm going to start with a few of my inspirations for molecular illustration. So the history of people, creative people drawing pictures of molecules is not quite as long as for natural history illustration or medical illustration, but just the same, there's 50 or 60 years of tradition of work by people trying to solve this problem of how to create comprehensible illustrations of the molecular world. And so I'll just show a few of the people that have inspired me over the years. And so I'll start with these two, which are collaborations between an artist and a scientist, okay? So over here on the left, Linus Pauling wrote a textbook called College Chemistry. And I got this textbook from my father. He used it in college. And this book is filled with beautiful line illustrations by Roger Hayward. Everything from beakers and retorts down to individual molecules. And they're such beautiful, tactile, you can just see these things right in front of you. It was a great way to be introduced to this world of chemistry. And so that book actually got me started with an interest in chemistry. Later on, when I was an undergraduate, I found a book by Irving Geis and Richard Dickerson, called The Structure and Action of Proteins. That kind of did the same thing for the field of protein structure. And so they got together when there were just enough protein structures to start thinking about some general principles about how proteins are built and how they function, and put together this lavishly illustrated book that walked through basic protein structure, a little bit about immunology, a lot about hemoglobin and myoglobin, and how they transport oxygen. And very much a collaboration between the scientist coming up with what needs to be shown, and Irving Geis coming up with all of the visual ways to make that clear to a wide audience. And so Irving Geis is actually the father of this molecular illustration, and everything has kind of built on him since then. And I'll just put a little shout out here that the archive of his paintings and sketches, which are amazing, has been archived by HHMI and the Protein Data Bank. And throughout this talk, I'm going to say PDB a lot. That stands for Protein Data Bank. The Protein Data Bank has gotten permission to put these online now. So they're building up this Geis archive bit by bit with the help of a bunch of undergraduate students to make these paintings available, so they're not just hidden away in the archive anymore. So if you're interested, I invite you to go look at that online archive and learn a little bit more about the seminal place he's played in protein illustration. Okay, moving on. When I was just applying for graduate programs, a crystallographer at UC Riverside handed me this paper by Jane Richardson called Anatomy and Taxonomy of Protein Structure. And so this was a seminal paper by a person, Jane Richardson, who is a combination of a scientist and an artist. And so at the time that she wrote this, there were, probably 40 different protein structures that were known, 40 or 50. And she developed this way of representing the way the protein chains are folded into globular proteins that's very clear and allows you to compare the similarities and differences between different folding patterns in proteins. And so revolutionized the way that everybody thinks about the way that proteins fold, allowing you to compare a bunch of different structures in a similar representation. And so this paper, for me, told me that I had to be a structural biologist, right? And then finally, I'll just acknowledge here Art Olson, who runs the molecular graphics lab at TSRI. I did postdoctoral work with him, and then just kind of stayed on ever since then. And the thing that he has brought to the representation of molecules is just a real exploratory spirit. Okay, so he was one of the first people to apply the brand new methods of computer graphics to representation of molecules. So this picture up here is one of his movies showing virus structure using a system that was brand new at the time, the Evans and Sutherland multi-picture system that allowed you to draw thousands and thousands of these beautiful glowing lines and then rotate them in real time. Brand new technology, nobody knew how to use. And so he made a number of movies showing the structures that were available at the time. He also developed some of the first methods for using raster displays to display molecules. For instance, this is a science cover that he drew showing a researcher that had synthesized by hand, chemically, an entire protein with the opposite hand of amino acids, and showed that it actually worked exactly the same if you gave it a substrate that was the opposite hand as well. So, interesting scientific experiment. And so now Art has kept this exploratory spirit to this day, and the kind of stuff he's working on in the lab now is applying 3D printing technology to create physical models of molecules, and then he's using those physical models in augmented reality as input devices so that you can play with the molecules and then overlay things like electrostatic potentials and stuff on top of that in the computer display. And then more recently, working on methods for creating 3D models of this cellular mesoscale of molecular detail inside entire cells. And so this is a picture from Graham Johnson, who did his doctoral work with Art, and a very talented program, Ludwig Ouden, who are today working on these methods for creating real quantitative 3D models of this mesoscale. So anyways, those are some of the inspirations. I'll just show a few of my early pictures here. These are a few things from my graduate work and postdoctoral work. And again, at the time, back in the 80s, we were kind of making it all up as we went, using the technology that was available and trying to come up with visual solutions for capturing different aspects of the molecular structures. So up here is one of those picture system images. You could draw lines and dots for this thing. So we did a nice bond diagram for the DNA here, some little dots to show how big the atoms were. And so this was meant to show how this is a DNA molecule and this is an anti-cancer drug that binds in the groove. So we were trying to show that it fit really nicely in that groove. For these DNA crystals, we were always obsessed with how the different helices pack into the crystal lattice. So here are a few hand-drawn things that I did for journal articles. This is an image from a pen plotter. I don't know if anybody in this room has ever used a pen plotter. It's a thing that would go and draw hundreds of little lines on a piece of paper. It's the best we had at the time. This is one of the first raster display images that I did. Really super slow. This image took an hour or two to calculate at the time, and then the screen was only 8-bit raster. So I would put up the red, open up the shutter of my camera, put up the green, open up the shutter, put up the blue, open up the shutter, and hope that it didn't move too much between the three. Disaster. And then in my postdoctoral work, I worked on some volume rendering algorithms, which are ways to take fields of data. This is electron density values, and they use these wacky glowing clouds to show that. And we spent a lot of time on it, it never went anywhere. Too hard to see what's going on, but it makes for pretty pictures. So everything kind of changed for me when I found this paper at a SIGGRAPH conference. And so this was a really nice little algorithm for taking 3D computational models and then generating these computer-based illustrations that look just like what you draw. Okay, nice outlines, little gesture lines, highlighting all the major features of the object. And so I ran home and implemented this for molecules. And the idea behind this is super easy. What you do is you take your model of the molecule and then calculate a depth map. So this is a ribosome here, and the dark areas are further away from us than the white areas, right? Super easy. And then you just take this image and you look for places where there's a big difference between the front and the back and draw a line. And so you get this. So it's exactly what we do when we pick up our pens, right? We're looking for the features that are the biggest things going on in the molecule. So I went a little bit crazy with it and did all kinds of wacky stuff with hatch lines and shading and all of that. And it kind of all settled down to the style I use now in the column, which is very simple. It's using these outlines just to give us a nice feeling for the overall shape and form of the molecules. And then a coloring scheme that's very understated. It's meant to highlight the different subunits of the complex that I'm showing. And I think the important part of these illustrations is that I want to be able to show the relationship between a molecule and an atom. So if you look carefully at these things, you can see how big an atom is, right? All these little bitty balls. But you're not getting caught up in carbon next to all the details. You can step back and see the overall shape and form of the complex and how the pieces are fitting together. I allow myself a little bit of change in the color and make all the carbons just a hair paler, just to help separate the atoms from each other just a bit. But not enough to distract from the overall complex. Okay, so that's what I've used for the past 10 years or so. These are digital pictures. And I love it when people ask that question, and later on with the paintings they'll say, are those digital? No, those are hand-drawn. So I'm very intentionally trying to come up with a style that spans this range, but uses the tools that are best for the particular scale range and the data that we have. So for these molecular pictures, we have atomic data. We have coordinates for where every one of those atoms are. So I want to use it. When I draw the cell pictures, I don't yet have 3D quantitative information for that, so I do them all by hand. I also just wanted to mention this little program, JSmall, which I use for everything now. It's a free, tiny little program that allows you to put together these nice little web interactives with buttons here to change whatever you want. And to me, the most important part of it is that it has this really nice natural language scripting ability. So you can say, select protein color red, and it does it. Or select liquid color blue. And so we very typically, we get high school students in, and we give them a half an hour of training on this, and they're running out and drawing pictures of molecules instantly. It's very simple and very powerful. I actually just, for my next book, figured out a way to write out nice large files from the thing, and they come out beautiful. So I'm actually using this for my publication quality images now as well. So a little shout out, if you're going to play games with molecules, look up this right away. It's fast and easy. Okay, I'll talk a little bit about some of the challenges of drawing pictures of molecules, and there are many. And so, 3 steps for this: choosing a structure, choosing a representation, and playing with colors, which is the fun part. And the most challenging part of these illustrations is finding an atomic structure to start with. Okay, and the reason for that is that all of these atomic structures that you find in the Protein Data Bank archive, this is the primary experimental data from researchers. Okay, so you have to deal with all the stuff, that, all the baggage that comes with that. And so a few of the challenges that you'll have, first of all, is finding a structure within an archive of 100,000 structures. Okay, so here's an example, probably can't read it, where I searched on the word hemoglobin, right? That was the first thing you want to draw pictures of. And it comes up with 687 different structures. And so now how do you find the 2 of those that are oxy and deoxyhemoglobin to tell your story, right? Another big challenge is that crystallographers will do anything that they need to do to get a structure. Okay, so very often you're going to end up with structures that are missing pieces that are moving, and the crystallographers can't see them. Here's an example of HIV protease, where the very first structure that was solved didn't have a substrate in the active site, so these 2 loops that close down over it and help position the substrate before it does the cleavage, they're flapping around, so the crystallographers don't see it, and so the structure won't have coordinates for that. What do? You have to model them in or something. And it wasn't until crystallographers solved structures that had something in the active site that these loops settled down and you could start to see them. So that's extra work. If this is the only one you have, you're going to have to do some modeling. And worse than that, you have to know going into this to look for the things that are missing. It's not always obvious from the structures which bits are missing. Other things like this ATP synthase, which is an incredible rotary motor in cells, is very mobile because it's a rotary motor. And so the crystallographers have had to chop this into pieces, solving structures of each of the rigid pieces. So you go to the archive, there's no structure of the whole thing, but there are structures of all the bits and pieces. So if you want to do an illustration for a textbook, you need to cobble these all back together. It's a big challenge. So the Protein Data Bank has a few resources to help you chart your way through this if you get a commission and need to do some molecular illustrations for someone. The first is that they've been working very diligently to improve the search capabilities on the RCSB Protein Data Bank site. So for instance, there's just a classic search bar at the top, which is getting closer and closer to a Google style thing, where it'll add some extra intelligence to help you chart a path. And then a whole bunch of features over on the side that show different classes. For instance, it'll tell you different organisms. These are some classic names. So for instance, down here, if you're looking for the hemoglobin that's found in legumes and protects them from oxygen, you can find the entry here for all the leg hemoglobin structures. And there are other ones for the size of it, whether it has proteins or DNA and all that kind of stuff. So a bunch of hints to help you chart your way through this. For people that are a little more sophisticated about it, there's also this nice new protein feature view that relates sequence information back to structural information. So this one that I've shown here is a particularly tricky subject. It's a molecule called the p53 tumor suppressor, which is super important in cancer, the development of cancer in cells. And it has 3 domains, one that affects the transcription of cancer genes, one that binds to DNA, and one that ties 4 of these chains together. And again, they're all connected by flexible linkers, so you're not going to find a structure of the whole thing. So it takes information up here from UniProt, which shows the sequence of the whole protein, and then these are the bits and pieces of the protein that are available in the Protein Data Bank. And so we can cobble together the whole structure based on that kind of information, picking an appropriate DNA binding domain here, transcription domain here, and tetramerization domain here. But it takes some legwork to work through it. And then one of my approaches to helping people chart a path through all this information has been this column that I've been doing at the Protein Data Bank for the past 15 years, called the Molecule of the Month. And the goal of this is to take a subject. The major goal of this is to take a subject each month, some molecule that's important in our health or welfare or industry, and then pull out a few structures from the archive that are going to be the best place to start. Okay, so here's an example of a sodium-potassium pump that pumps ions across cell membranes. And so I pulled out one structure for the sodium-potassium pump, a little secondary topic that shows some related pumps, and again, saying this particular entry is a good place to start. And then down here, a section called Exploring the Structure that talks a little bit about the structure-function relationships, and maybe shows you the way to start telling the story about these molecules. So I've done about 200 of these columns now. So we have most of the major topics that you'd be asked to do if you were going to do a textbook. Okay. So that's hopefully a good place for educators to start, and I hope people in this room to start if you need to tell these kind of protein structure function stories. And so a number of years ago, Helen Berman, who was the head of the PDB at the time, compiled all of these different columns I've done into a resource that she called PDB 101, and put some nice browsing tools together to allow people to, for instance, if you're telling a story about diabetes, to pull up all the columns that are related to that, or all the columns that are related to cancer, or different topics like that. So this is a good place to start if you're tasked with telling some microbiology stories. Okay, so, is that for me? Okay, so after that whole process, which is actually the most challenging part of the whole thing, once you have your structure in hand, you have a few decisions to make. Okay, so when you're creating these representations of molecules, you're going to try to capture some aspect of the molecule that's important to the story you're telling, it's important to the function. Okay, and you're going to throw a whole bunch of other information away because the images just get too complicated if you're trying to do everything at once. So down here, my favorite representation is one developed by Linus Pauling 100 years ago when he was looking at basic chemistry, the space filling representation. So you're just putting a sphere at every end. And I love these because they kind of give you an impression of what it would look like if you had it here in your hand. You know, the size and the shape of it, how they fit together, and all of that kind of stuff. The one shown here is this is a circadian clock protein. And as part of this function, it has this little tunnel that goes down through the middle. And the nucleotides can only reach it from the inside. So the space filling is a great way to show that this is a thing with a tunnel down the center. If you want to tell more detailed stories, you can go to these kind of wireframe pictures that show the position of every atom, how they're all bonded together. But I've found for that kind of illustration, you need to really focus on one particular little aspect. So for instance, this protein that links up in an amazing concerted reaction, this minosterol molecule forms a whole bunch of bonds all at once to form a cholesterol precursor. And so for this, the story I wanted to tell was that reaction of the formation of the cholesterol bonds. And the rest of the protein, I want to show that it's there, but I don't want to get mired in that detail. So it's a very simple backbone representation, just to show that it's kind of all happening in the context of the protein. Those kind of backbones are also great for telling stories where we're comparing two different structures. for instance, if you overlap these two and have people flip between the two. So this is showing that in this phototropin molecule, that most of the protein doesn't move a whole lot. Just this one tiny little cysteine here is doing all the work when it goes to the light-activated versus the dark form. And in the molecule, in the interactive, I have those two overlap so that people can flip back and forth between the two. And then down here, Jane Richardson's ribbon diagrams are an essential tool if you're talking about protein folding, and also just for sheer wow factor. You know, these are going to be the most beautiful way to represent a protein. Maybe not always the best way to show the function you're trying to show, but a way to create a really beautiful illustration. And they're used for that a lot. Actually, they show up in a lot of editorial kind of applications. Okay, so finally, after you've chosen between all of those different things, you get to do the fun part, which is to pick some colors. Down here are three of the defaults that are going to show up on any computer graphics program that you use to draw pictures of molecules. Here's the classic coloring by atom. This comes directly from Linus Pauling, from his 3D models. Oxygen's in red, nitrogen in blue. So every chemist in the world is going to understand what's what in that illustration. So really great for scientific publications, because you don't have to do a lot of explanation. Scientists are going to understand. Here's a coloration by subunit, just to show that hemoglobin is composed of 4 different subunits that are similar. And then a representation here, coloring by rainbow from one end of the chain to the other. And this is a nice way to help people follow the way these chains go up and down through are folded into the overall structure. Used quite a lot. And then up here at the top is the scheme that I've used most often. Coloring by subunit, and then using just these subtle colors to differentiate between the different type of atoms in the structure. Okay, so I'll move on from molecules to cells now. We're doing halfway through, good show. Okay, so this second goal that I've had over the years is to create illustrations of the cellular mesoscale. So this is the scale level between molecules and ultrastructure of cells. And so this is a really, really exciting place to work, because there's no experimental way to look at this directly. Okay? We have a bunch of wonderful techniques like X-ray crystallography to look at the detailed structure of individual molecules. But when you do that, you have to purify the molecules away from their cellular context and just look at one of them. So you completely lose where they are in cells or what they're doing in cells. At the other end, we have microscopy, which can show us the shape of whole cells, where the membranes are in them. But until very recently, the resolution of microscopy has not been enough to resolve individual molecules in those cells. And so in between that, we have a bunch of techniques. Basically what you do is you grind the cells up and you count how much of what you have in the goo that's left over, right? And hopefully you can retain some of the molecules that are interacting in that goo. And so these proteomic models can help tell us how many things need to be there and sometimes where they need to be. And so what I try to do is combine all of these types of information and synthesize a picture that shows us what's happening at this intermediate level of a significant portion of a cell seen at molecular detail. And so the goal of this, I'm trying to create a picture that shows all the molecules, where they are, at the proper size, interacting with their proper partners, as much as I can based on the available data. I have a few goals for these when I use them for educational outreach. These are a few very simple goals. The first thing I'd like to do with them is to relate cells to molecules to atoms, which is not a clear thing to a lot of people. And it turns out you can do that in 3 steps, 3 visual steps. And so these are some illustrations from my book, Machinery of Life. So up here is a picture of a whole E. coli bacterial cell, okay? And then you can box out a tiny little portion up here around the flagellar motor, which is distinguishable at this magnification, and enlarge it here. So here's that flagellar motor that we're seeing right up here. This is enough to show individual molecules, right, inside that one cell. And then we can take an individual protein in that. If I can see it, this little butterfly-shaped one here, phosphofructokinase, and you can blow that up to the place where you can see individual atoms. So 3 steps going from a cell, a small cell, down to an individual molecule. And you can see the atoms in this as well. Another thing I try to do is to show different functional compartments in these cells. And you know, it'd be nice to be able to use color to do that. But there are only a few ways to parse that out. The way I've chosen to do it is to show the overall structural features. For instance, the membranes here in green and this nucleoid region in yellow in the center. But unfortunately, there are also a bunch of different functional things happening in this cytoplasmic region. And so in the book, I chose to use a number of key figures to call out different functions within that space, rather than coming up with a coloring scheme that tried to show both the structural features and the functional features in the same figure. It just would have gotten to be a mess. So for instance, here's a figure that shows all of the proteins involved in energy metabolism from this space right here. And in that same space, the second figure calling out the proteins that are involved in protein synthesis. So probably difficult to show both of those kind of features in one, by coloring in one illustration. But whenever possible, I also like to try to tell biological stories in these illustrations. So here's an example of a synapse. And so for this, I tried to show a whole synaptic cycle, the process of the synaptic cycle, starting with a vesicle here filled with neurotransmitters, docking to the surface of the cell. This would be the axon. It binds to the surface of the cell and dumps the neurotransmitters in here. They diffuse across to the muscle cell, binding to acetylcholine receptors here. And then finally, they're broken down by this acetylcholinesterase after the signal is finished and transported back into the muscle or the synaptic cell by this tiny little protein here, which is a pump that squirts them back into the cell. Another thing that I like to show with these is that there's a lot of infrastructure that's involved in this. So if you think about it, the only thing that's really important for the function of this whole thing is this neurotransmitter and this receptor cell, right, the receptor protein. All of this other stuff is there to hold everything in the right place at the right time so that the neurotransmission can happen at the proper time. So we have all this infrastructure inside the muscle cell to hold the acetylcholine receptors in the right place. We have all this infrastructure to store the vesicles, make sure that they're transported to the surface at the proper time. And there also is a bunch of regulatory information down here that helps make sure that this is all happening at the right time. So a lot of infrastructure to deal with here as well.
Is this a watercolor?
This is one of the watercolors, yeah. And I have a little thing later on where I show the process of putting one together. So.
Can you just say how big that is, the original?
I draw them twice the size that they're printed. So it's, and they're drawn at 2 million times magnification. That's a big help, isn't it? The advantage of that is, of course, that I can put all my source materials together and do picture after picture after picture and propagate all my errors. Okay, so yeah, so since this is an audience of kindred spirits here, I'll talk a little bit about some of the artistic stuff that goes behind all of these. So I made a whole bunch of choices when I started doing these pictures. So at the time, the most common way people would draw these, first of all, would be not nearly as dense as they should be, right? And then the animations of the time show this sparse cellular environment, and then the proteins would sail through beautiful astronomical celestial bodies. And as part of that, they were always kind of an immersive view. You know, you were a submarine and these things were flying around your head. And so very early on, I chose not to use that kind of immersive approach. Instead, I'm drawing this like, take a cut through your cell and you're holding it right here in front of you, right? So it's all orthographic projection. And I think the real advantage of that is that I can show big fields of these molecules, and also it allows people to go and compare ribosomes to DNA, to flagellar motors, to this little endotoxin protein. So they're all in a consistent representation with a consistent scale. So you can keep all of those relationships in mind without having to worry about, well, that one's further away, so it's going to look smaller, and this one's closer, issues like that. So the images are meant to be a cross-section that's really rather shallow, drawn in orthographic projection. There's a lot of artistic license in the molecules that I choose to show. Based on proteomics, I put all the most common things, like all of these different ribosomes are at the proper concentration, and then I fill up all the spaces with everything else, everything I can think of, just so I can tell a fun story. So for instance, there probably wouldn't be a section this big that includes both RNA polymerase making messenger RNA and a DNA polymerase replicating the DNA and a repair protein repairing the DNA. That probably wouldn't all happen in a space this size, but again, I'm trying to show a lot in this one picture. There's also a lot of artistic license in the way I do the cross sections. So for instance, this DNA tangle down here, these yellow guys are the DNA strands. They should be coming all different angles, right? And so there should be a bunch coming right at me that would be clipped. And so the first pictures I did a lot of that clipping, and it was a mess. So now I allow myself the ability to put all of the DNA in the plane of the page, okay, so that it reads better as a big tangle of one big DNA circle. It's wrong, right? And as we move to 3D modeling for these and try to start asking scientific questions, that's going to change, right? It's going to have to be a real 3D model. But for this application, I think it's okay. Using these very flat colors, simple outlines as a way to let people see the whole scene instead of getting all caught up in the tiny details of each individual molecule. But using little details like this to let people explore individual molecules in more detail, but not in the landscape pictures. I don't want that much detail because then you get lost in the overall interactions. What else? Captured the flag. Okay, that's about it. So I'll just walk through a few pictures to show how I put these together. And so this little quote is, I think it was from the last GNSI conference I went to. I had this painting. It was one of my first watercolors. I was very proud of it in the show. And somebody came up to me and said, so after you draw the lines, it's all paint by number, right? Of course, I was forced to say yes. And actually, it's even worse than that because I actually do the paint by number first, and then I draw the lines wherever I happen to have painted it. So, but there you go. It's a style that I've developed that allows me to get through these paintings in an amount of time that doesn't drive me insane. So I'll just walk through the process of putting one of these together. I recently did a a series of pictures of the HIV life cycle for a book on the current research in HIV biology. And so I'll just walk through the one that shows the protein synthesis. And so, of course, half the time I spend on these illustrations is going to the library and reading about the science. And so these are some of the pieces of information that went into the picture. The most important thing is that the HIV genome forms this tiny little hairpin that stalls the ribosome 80% of the time so that it makes a short protein, and the rest of the time it jams its way through that and makes a longer protein. So you get 80-20% of these two different short and long proteins. And then I also found some references to show that the polysomes these days, people think of them as being, so there's a long messenger RNA that the ribosomes zooming along and making proteins, but the two ends of it tend to be associated with each other to form a big circle. Who knows what that's for? But electron microscopy has support for that, so I wanted to show that, and then a whole bunch of individual structures to show how these ribosomes get started. There are a bunch of proteins that find the start of the ribosome, get it started in its protein synthesis. So the painting, those were the basic pieces of information that I wanted to show. I wanted to show the hairpin, I wanted to show the circular polysome, and I wanted to show a bunch of that initiation complex, since it's kind of current information. So I start with a big sketch to scale, like I said, 2 million times. You can see this is the initiation complex, and the ribosomes kind of circle around here. The hairpin was down here at the bottom. Circle up here, circle, 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 and then the tail is right here. So the sketch, this is where I work out all the problems. Basically drawing in the major players first, and then filling all the wiggly RNA in between those. This is transferred to watercolor paper with this carbon paper my grandfather gave to me 40 years ago. Fortunately, it's an interesting self-healing carbon paper, so it's still working now, but when I run out of it, I'm doomed. And then it's just a very, very simple process. Watercolor palette that my grandfather bought for me, and a very tiny brush, throwing stuff in. So here's the process of that foreground, what I'm drawing. The RNA first, filling in the ribosomes, filling in the red viral proteins that are being made by the ribosomes. At this stage, all of that foreground has been drawn in. And then I kind of just extemporaneously fake in 2 more layers behind that get progressively darker. And this is the point when I almost always just want to give up and throw the thing away, because you lose all feeling for the tonalities, colors. It's just a mess. I just have to trust that it's going to work out. Right? So here's a few darker ribosomes showing up behind here. This is with that whole second layer behind filled in. I generally just use tracing paper, trace the shapes of the molecules and then dot them in. If I used acrylics or something, this whole process would probably be a lot easier, because I wouldn't be filling in all those little spaces in between every time. But my grandfather started me on watercolor, so I stuck with it. So here's the third layer put in, and then just these tiny little white chips filled in with black. And then the final thing is just to clean up the whole thing with a bunch of black outlines. So up here is that initiation complex, some little hairpins that are in the HIV genome that are important for regulation. It winds around here, the protein is getting longer and longer and longer. Here's that little hairpin that stalls it. More ribosomes with the gag-Paul protein getting longer and longer. There's a whole bunch of RNA there that's involved in making a bunch of accessory proteins. I couldn't find any references to tell me what actually that looks like, so I kind of just put it up on the side. And then these are These are the poly-A binding proteins that cover the tail of the RNA, interacting here with that initiation complex. So that was the final picture for the book. So I'm running out of time. So yell at me when I have to stop, okay? Good. Now, the rest of the talk just has a bunch of recent things I've been doing. I'll just dash through a bunch of these. Okay. So here are two of those illustrations from that HIV book showing the process of reverse transcription, where the RNA genome is transcribed into DNA. So this was the first version that I did it, showing the capsid here from HIV, the RNAs in yellow, and some little green strands of DNA being made on that by the reverse transcriptase, which is the enzyme here. And so I showed this to a bunch of researchers in our center, and they all go, "This is all wrong." So it turns out that there are no publications that say this, but the current fashion is that all of this is happening inside the capsid, which makes sense because it kind of protects it from all the enzymes in the cytoplasm that would break the RNA up. So I quickly went back and redrew the whole thing so that the reverse transcription is happening inside the capsid now. So this is the one that went into the final book. There's also evidence over here that this dyning motor crawls down microtubules and drags this thing down to the nucleus, and this is totally made up. This is the most recent one that I've done just last month for the same center, just incorporating a few pieces of new information on the structure of HIV. In particular, the fact that these enveloped glycoproteins, which are the things that stick to the cells, that they in fact tend to be clustered in one place instead of evenly distributed around the thing, which makes it so that it has a bunch of different points where it's interacting with the cell. And also some brand new stuff showing that the integrase protein here condenses the RNA into this tight little ball inside the capsid. This is brand new information. So as part of this center grant, I do pictures once or twice a year to update everything based on what the most current information is from the science, which is a lot of fun. Learned some big lessons with these two pictures, which is draw pictures of viruses that are in the news, and you get in the news. So, you guys know what these two are? Any guesses? This one, Zika and Ebola. Yeah, so I did both of these for the column at the Protein Data Bank, and picked up a lot of press for them because everybody wants pictures of viruses that are in the news. This was a really fun project that I did with Tim Herman at the Milwaukee School of Engineering, and he put together a project, NSF-funded project, that brought together students, I think they were undergrad students, a researcher, and his team of educational curriculum developers. And together, this team put together a story, some molecular story, that related molecular structure to function. And so this researcher worked on proteins that were involved in the pinching of vesicles inside of cells. So they brought me in, excuse me, they brought me in to draw a picture of the whole process, and then the students put together this really nice interactive viewer that allowed you to go and click on different molecules in the painting, and then it would pull up the molecular structure, and you'd be able to rotate it. The students wrote all this great text for each of the proteins. It was really exciting. So they put this online, and a researcher contacted me a few months later and said, "This is all wrong." So this is one of the exciting things about this field, right, is that everything can be wrong. It shows that it's exciting, new, cutting-edge research. So this this illustration was based on a review, an illustration from a review article from the most prominent scientist in the field at the time. So I always try to find that kind of provenance for pictures, right? And so this woman calls me and says, well, we think that the dynein, which is the motor that pinches this off, and these bar proteins, which help form the curvature of the membrane, the kind of banana-shaped proteins that all line up along the membrane, they're reversed. They need to be the other way around. And I said, okay, give me some references to show that that's the case, and I'll do it. And he said, well, we don't really have any publications. So, so I said, okay, I'll draw it any way you want, for your publications. So I worked up this one with him that had the two reversed. He was one of these collaborators that we went back and forth on these actin filaments over and over and over and over and over again. It's the kind of person that would just say, no, try again. Please draw me a picture, please, please. But we finally got to a place where he was happy with it. And then I made the mistake of saying, well, it can be any color you want. So here are a few pictures from my book, which is a new book, which is coming out later this summer, which basically builds on the Molecule of the Month column, showing a bunch of, I'm out of time. Let's do, just two little things here, which is a fun project that we've been doing, which are trying to come up with activities that we can put on the PDB site for students to use, right? Activities you can do in the classroom to get people excited. So we've been doing these little virus fold-ups, one for Zika virus, and a little preview of what's coming out next month for my 200th column. It'll be a whole exploration of viral quasi-symmetry, which is a way that subunits of viruses are used to build larger and larger and larger capsids by allowing some deformations of symmetry. So I'm making a whole series of things for different sizes of viruses. So you might take a look at that. And I guess I'll finish with that and take some questions.